protein structure and protein sequence and structure relationships and the search of protein function into protein structure. At a certain point, I didn't really leave proteins, but was somehow very strongly attracted by RNA. And today, so I decided to tell you this story, the story of our involvement and of the importance of RNA in biology and in computational biology. Let's see if the presentation works. Yes, it does. So a brief outline. I will start summarizing why we decided to work on RNA, why RNA can be considered so interesting and full of possibly surprising new things that we can still discover about its biology, and how it is possible to study and analyze RNA molecules. And then I will talk about the tools we have developed to analyze RNA. Because, as I will say along this talk, there are very many tools already developed in the years for protein bioinformatics and computational biology. For RNA, there are not that many, not that bright, not that good. And so we decided that more was needed to really understand the role of the new role that RNA is taking on the scene in biology in the last 10 years. And then I will describe the application that can be done with these tools. So first of all, so why RNA? As you see here, we have the stars of the molecules in cell life. And let's start from DNA. DNA, as you know, is generally a stable double helix, and it stores genetic information. This is in general its main role. Of course, sometimes can also be seen as a single-stranded molecule, but most of the times it is very stable and it encodes all the information that is necessary to start a new cell. Then proteins. There are very many proteins with different architectures, different roles, different abilities. They can have just a structural role. They can transmit information, transport information. They can bind small molecules or other proteins, and they can also be enzyme. So through the reaching their final 3D structure, they can have roles: cutting, binding, recognizing, informing proteins. And RNA, between them, what is it doing? RNA, as you see here, is generally, again, a single-stranded molecule. And when I was in my infancy, when I started studying molecular biology years and years ago, RNA was only the molecule transporting information from the DNA to the translational machinery, so allowing the genetic information to become flesh and blood, to become proteins, which were the actors of the work to be done in the cell. But RNA is much more than that. As you know, can also be able to store genetic information, like in the case of our not beloved friend SARS-CoV-2 virus. There are several viruses in which RNA is the genetic molecule. And also RNA can fold into nice and complicated 3D structure, a bit like proteins. It's a linear molecule which finds its lowest energy somehow folding in a structure. And more than that, this 3D structure can also have enzymatic functions. They can cut, they can bind. Also, under this respect, they are very similar to proteins. And So RNA is a very versatile molecule. And here in the figure, you see the case of RNase-P. This is a protein RNA complex, which is quite conserved from prokaryotes up to eukaryotes. And this complex is the one who cuts the 5' of tRNA genes. And what has been discovered is that the enzymatic function is totally mapped into the RNA. The protein just helps, but the protein alone cannot cut, the RNA alone cuts. And not only, for sure, we all have studied the big role that RNA has in translation, being the majority of a ribosome, also in the formation of the peptidic bond, which is so important to build the proteins. The RNA has the enzymatic role. If you try to work with a ribosome depleted of proteins, nothing happens. If you work, depleted of RNA, nothing happens. But if you deplete the ribosomes of the proteins, then, of course, much less efficiently, but peptidic bonds are formed. So RNA can have very different roles. And for this reason, probably, it is thought to be the molecule with which life started on Earth. So now what you're seeing is the case of the human genome. It was already known that only a relatively small part of the human genome encodes the information for proteins, from 1.5% to 2%, I think. And that there are very many RNA genes that were believed to be just a sort of dark matter in the human genome. Not really useful, just, like I am an RNA polymerase, I have to work, so I transcribe. And this was a little bit odd, but somehow accepted that this RNA was useless. With time, it has been discovered that this is probably not true, or not 100% true. Let's say maybe better and more seriously. But at the moment, these are the numbers, the figures I took yesterday evening. In the human genome, there are 20,400 protein coding genes and 24,000 non-coding genes. Of course, in these non-coding genes, there are very many genes, such as the tRNAs, the ribosomal RNAs, the microRNAs, and the RNAs, the small nuclear RNAs, which are involved in translation and in control. That's it. But there are also very many of these genes which are not only transcribed, but spliced. So they have introns and exons, and they are spliced. So this means that probably they are useful. They are capped, and the poly A tail is added at the 3', and they work. For a handful of these non-coding RNA, we have a function. But for the great majority of them, we really have no clue about what they do. And the more they are studied and associated, they are associated also to diseases, to different functions. But again, at the moment, very, very few RNA are known, non-coding RNA are associated to a known function. And so what are the non-coding RNA functions that are known at the moment? As you see in this picture, can you see my mouse?
Yes, I can see it.
Yes.
Maybe because when I have students in the lessons, they always ask me to change the mouse in this way.
Okay.
So maybe for some of the audience, for part of the audience, this is better. So as I was saying, these non-coding RNAs can work inside the nucleus and in the cytoplasm. And they are known to be able to share different roles. As you see, they can be involved in the recruitment of chromatin modifiers. This means that they are involved in the regulation of transcription. They are part of the transcriptional machinery. They can bind to promoters. Now it is known that they can bind also to enhancers. And this means that they can change, that they can activate or repress transcription in eukaryotes. What is known since very many years is that they are involved in the splicing. And since the 3D structure of the genome in the nucleus is analyzed in the last years, now it is known also that they are involved in this structuration of the genome, in the formation of the TADs, which are conserved and associated to the story of what happens to the genome from a metaphase to another metaphase. And also, they are involved in the formation and function of different subnuclear structures. Then what they do in the cytoplasm, of course, they work in translation. MicroRNAs are known to be able to repress the translation of the target's messenger RNAs, or also to decide about the fate of a transcript, digesting the different transcripts. Long non-coding RNAs can be used as sponges, so that different microRNAs can bind using the normal Watson and Crick pairing ability, and so they are depleted from their presence inside the plasma, the cytoplasm. So about non-coding RNA, this is just an idea of what it is known, but for sure, what is thought is that structure is important for function. And from the analysis of the different genes which are present in the different genomes doing probably the same thing, it is known that the primary sequence tend to diverge faster than the structure. This is true also for proteins, so we can handle this problem. But when you work with proteins, you have 20 different amino acids. When you work with RNA, you have only 4. So comparing sequences and having to handle this divergence become much more difficult. Because when sequence identity between two RNA molecules drops below 60%, then you don't know if this alignment is significant or if it's nothing. And in this respect, structural information becomes essential. So as I was saying, computational biology has produced very powerful tools for the analysis of protein sequence and structure, but with RNA, much less, because RNA was not thought to be so important, because non-coding genes are on stage only in the last years, only recently. So let's think a bit about the problems and the needs that we have about analyzing RNAs. In this case, we have an alignment, a high sequence identity. So we think that this alignment is significant. In this case, we don't know, but we think that this alignment probably is not significant. Looking only at sequence identity, we have to stop here in our consideration. Now let's look at these other two molecules. These two molecules share a very low sequence identity, if any, but let's look at these two different sequences. They can fold into a secondary structure using the Watson and Crick pairing. And look, these two secondary structures are very similar, if not identical. And more importantly, in this secondary structure, the same sequence is exposed in a loop. So this example shows us that a proficient use of sequence and structure information can help a lot in the analysis of non-coding RNAs. For example, it is important to know that in many cases, the interaction between proteins and RNAs happens involving RNA stems. While when RNA binds another nucleic acid, another RNA or DNA, for instance, for a regulatory scope or role, then the interaction usually involve its exposed residues. So this ability to handle this information is very important. What I wanted to tell is that, for instance, examples of this. This is a stupid example that I designed at a certain point to communicate this information. But of course, everybody knows that introns, for instance, can fold into a specific structure and expose specific sequences, which are recognized by the splicing machinery. And both features are important for the splicing to occur. And another case that it happened to me to study when I was a molecular biologist many years ago was the regulation of the CoLU1 mechanism of regulation of DNA replication. In this case, again, this regulation was on the shoulders of two RNA molecules, and their interaction started from the loops, which were complementary, and then was helped by a protein that was binding to their stems. So for sure, there are also very many other cases still to discover. And now what about RNA data? Of course, in the last years, also thanks to NGS technology, we are flooded with primary structure data. So we have very many sequences, gigabytes, billions of bases, and growing. So the sequences for sure are not limiting. About structure. About structure, the situation is less happy because first, a lot of structures which are known structures are more or less the same because they are tRNAs and they are ribosomes. Very interesting, but redundant. So the information, for instance, yesterday I was listening to the very interesting talk by David Jones. This method that he and Google and others worked on to have these beautiful new predictions of proteins, of 3D structure of proteins. There's a lot of information to feed this neural method. In the case of RNA, I think it is nice. Maybe we can go on working for another while before Google arrives and solve all the problems for us. In our case, there's not much information about RNA. Of course, there's information in multiple sequence alignment, but this the need to jump onto the structure is always there and is much more difficult. So what is the structural information we have really? So this is information, again, yesterday evening, I took this picture in the PDB site, and these are 9,000 protein nucleic acid complexes. Only a part of these complexes, of course, are with RNA. And these are only nucleic acids is 3,500. Looking only at RNA, as you see, we have more or less 1,500, 1,500 known structures, which is not much. It's not much even because we have talked about this high level of redundancy that we have into the database. There are also riboswitches represented, but about what about 5' UTRs, 3' UTRs, or other structures which for sure are involved, are recognized by proteins and are involved in the regulation machinery. So we have this tremendous gap about the number of primary sequences we have, and the very low level of information we have about structure. So let's think. Let's think to secondary structure. What is secondary structure? In secondary structure, what is thought that what you see is the strong interaction that is anyway present into the 3D structure. And differently from 3D structure, even if a lot of work is being done, but again, the prediction of 3D structure is not, it still needs infancy. About secondary structure, there are advantages. You can have as many secondary structure as you want using the predictors of primary sequence. And therefore, even if the prediction of secondary structure is not perfect, it is not perfect, as I will tell you better, at least you have a big database. You have the whole database. Now, this only, I can also switch. No, sorry, I wanted to show, but how to make it start? Oh. I have problems with this screen. Okay, so I'm sorry. I wanted to show you a nice animation of an RNA molecules which finds its final 3D structure, going to trying to find its lowest free energy. So how can be calculated? How can be evaluated, predicted an RNA 2D structure? I'll show you a couple of tables where experimental data have been taken. These are thermodynamic measures. Now I lost aiuto. Okay, I don't have. Okay, I'm back. Okay. Sorry. Okay, as you see here, these are pairs of nucleotides on one strand, on another strand. So when RNA molecules are paired in a double strand, for instance, in stems, two different sequences paired are associated different delta Gs. And they have been evaluated experimentally, thermodynamically. So we have these numbers. And this can be used to calculate the stability of a stem using its sequence. Of course, stems tend to stabilize a 3D structure, while the open structures tend to destabilize the structure. And the more they are long, and the more they destabilize. So also in this case, we have a table with delta G. In this case, these are positive values that are associated to the different length of a possible loop. So, for instance, in this case, this secondary structure, which appears to be quite stable, has a certain stability that can be calculated as with its delta G, which is the composition of these two values, the negative values giving stability in the stem, the stems, and the positive values coming from the unpaired residues. As soon as the result is negative enough, we have a good prediction for a structure, for a secondary structure. This is not the only method at the moment used. It's another important method is based on covariance. But okay, talking about covariance could take hours. But anyway, there are other methods quite useful and strong, but none of this method anyway is perfect. They tend, this method, to take in input the sequence of an RNA and giving usually one secondary structure in output. And this secondary structure can be the so-called MFE structure, which is the minimal free energy structure, which is calculated somehow by the different methods available to calculate it. This doesn't look too complicated, but the living cell adds more layers of complexity, unfortunately, because, as you can imagine, an RNA looks for its secondary and then tertiary structures, also according to the context in which the molecule lies. So, for instance, in a cellular context, it interacts with other molecules, and interacting with other molecules has an effect and an impact on its secondary and tertiary structure. And as I was saying at the beginning, RNA tends to be an unstable molecule because it has a 2' OH, which tends to digest and cut itself very often. And generally, the cell tends to digest the RNAs just to be able to drive new molecules according to its needs. So a way to preserve the RNA molecules from being digested and destroyed too soon is to modify its nucleotides. And modified nucleotides, again, have an impact on the structure of the molecules. And cations also are usually very nicely bound to RNA molecules and have a big impact on their structure. But not only, also RNA concentration, temperature, pH. So just looking for the minimal free energy structure doesn't seem to be the solution. It's too simple and it's too simplistic. So we have to be very careful. The number of structures with similar energy grows with the length of the molecule. So how many problems we have if we want to work with the secondary structure of RNA. So I gave you the bad news. Now I try to give you the good news. There are solutions. For instance, the reliability of 2D structure prediction can be increased thanks to different techniques which have been implemented experimentally. As you see here, there are methods working with the base selective probes. For instance, exposed A's can be modified, or A's which are in stem can be modified with different drugs, with different, I don't have the English word now. Solvent accessibility can be probed, and local nucleotide dynamics can be probed, and nucleotides which are neighbors can be identified. And with this information in hand, the reliability of predicted structure can be made better, can be increased. And I show you, there are these different techniques which are a bit noisy in their answers, but they are really very powerful because they can be used both in vivo and in vitro, and they can give answers at the nucleotide level. So now I'm showing you 3 different predictions of the same molecule, which, what is it? This is, I think, the 5' domain, yeah, of E. coli 16S ribosomal RNA. So this is the list of errors. The red axis are the errors. This is the list of errors which are done without using the constraints that now can be extracted from experimental data. This is the prediction when you add the data from the conventional chemical mapping, and this is finally the best possible solution that has been found using the SHAPE and pseudo-free energies data. SHAPE stands for selective 2' hydroxyacylation analyzed by primary extension. And that is an accurate method for probing RNA secondary structure at the nucleotide level. So the good news, but I'm sorry, this one shown in this picture is not a good news, but this picture shows us that very often, but as I already anticipated in the preceding slide, the minimal free energy is not the one that is used in the cell. So what is showing you in this, what I'm showing you in this graph here, you have delta G. So lower delta G are more stable molecules. And here you have an evaluation of the structural difference between the molecules. So as you see, one can have different minima calculated, but the biologically working molecule is not always, not often, the one with minimal free energy. So, to these structures, I've told you about the pros and cons, but I remember I remind you the big pros. That is that working with secondary structure, we can have data about the whole transcriptome, for instance, of a cell. And it is true that we can have fakes, we can have bad predictions, but each prediction can come with an evaluation of its ability, of its stability, and this can help us, for instance, selecting the top of the iceberg and only the most stable structures, the ones having the higher probability to be true. So, having a 2D structure, what can we do with it? So we can calculate the 2D structure for many molecules and give answers about evolutionary relationship working on a global alignment between sequences. So you see here an alignment between 2 RNA sequences, and one can also compare the secondary structures that can be calculated from these 2 sequences. So you can have the sequence and the secondary structure alignment, and having both, you can predict, you can infer a possible evolutionary relationship about these 2 molecules. And these structures can also be used to perform a local alignment. And the local alignment can be very useful to be able to identify in a great amount of different molecules, to spot possibly the structure motifs which retain and have a function. Now, how can we describe secondary structure? Because, okay, I've shown you slides with pictures, but if you have to give a secondary structure to a computer, since we are computational biologists, we tend to develop a method working in a computer. What can we give to our method? So you see here one of the most commonly used descriptors. This is the dot bracket notation. It's composed of only 3 characters. And as you see here, you have this secondary structure. This is the sequence. And brackets show the residues which are bound in a stem, while dots show the unpaired residues. I show you here, we have this GC pair, which is at the beginning of a stem here. Now we have these 2 A's, which are at the beginning of this loop here, and so on. This descriptor is useful, but it's not very informative. While this other descriptor, for instance, a tree-based encoding, the representation of a secondary structure with a tree, is much more informative because, as you see, the description is much more realistic and retains a lot of information, much more than a simple single nucleotide character. The topology is encoded in a graph, and since you have a graph, you can use as method for comparison the method already developed in the years in informatics, graph and subgraph isomorphism. They work very well, but they are incredibly heavy from the computational point of view. So you can have representation which is rich of information, but as often happens in bioinformatics, having retained information usually means that you have to spend much more energy in the computational side of the problem. So we decided to work on the fact that 2D structures are composed of different modules and can be thought as a modular object. So you see here the different modules, for instance, can be stems. A stem is defined as a classical base pairing between complementary residues. Then there are bulges, as you see here. Bulge is a short interruption of a base pairing in only one strand of stem. You can have hairpin loops here that occur when there are residues at the end of a stem that cannot pair between one another. You can have internal loops, which are unpaired residues inside a stem. You can have multi-branched loop, which are loops from which different structure departs, and pseudo-knots, which are quite difficult to analyze. Still, they are present in biology. So we decided to simplify the problem and decided to develop an alphabet for the description of RNA secondary structure. First of all, we decided to restrict our descriptor to only 4 types of secondary structure element, which are the stem, the loop, the internal loop, and the bulge. And we associated a different character, a single character, to each one of these type according to its length. So this is a stem of one only pair of residues, one character. This another character, and so on for each one of the structure you see in this picture. So what happens when you have this secondary structure with this sequence, which has this descriptor as dot bracket notation? In this new alphabet, you can transform each nucleotide, you can associate each nucleotide with one of these characters. A 5 residue loop, one character there. 3 residue stem, again, here, and so on, right? Bulge, okay. As you see, a secondary structure can be transformed in a string of characters, and differently from the dot bracket notation, these characters retain a lot of information. So the bare alphabet can describe the secondary structure as a string of characters, but it is more informative than the classical dot bracket notation. With this alphabet, the comparison of 2 secondary structures is reduced to the alignment of 2 sequences. So fast. You can use heuristics for the comparison when you have to compare transcriptomes, for instance. So this is interesting in a way. And more than that, since we came from the protein world, we decided that if we translate a sequence, a primary sequence in a string of characters related to secondary structure, then we can also think to define a substitution matrix for secondary structure elements. And how we did? We started from our FAM and retrieved multiple alignments of homologous RNAs, their sequences, of course, and we extracted from our FAM the consensus secondary structure from for the family, which is usually reported under the multiple alignment of the sequences. Then we converted the multiple sequence, the alignment, the multiple alignment of secondary structure in the new encoding, and then computed the mutation rate for every position in the multiple alignment, as can be done for the residues, for the substitution matrices which have been defined for the protein world. Okay, this is crazy. This is possibly a good idea if it works. Let's see if it works. So to see if it works, if it worked, we decided to try and different matrices. So we defined a positive diagonal matrix. So all identical bare characters are given the same positive score. Or positive group matrix, where all bare characters denoting the same type of secondary structure element have the same positive value. For instance, here you can have all the stems independently of their length, all the loops independently of their length, and so on. And then we randomized the calculated matrix per row and randomized fully the matrix we had calculated from our FAM. Each one of these matrices was evaluated for its ability to reconstruct a standard data set of alignments. How? You see here the comparison. We have the different matrices which have been used with reference alignments, a high number of reference alignments. In this reference alignment, each couple of molecules had a certain number of residue pairs. How many of those pairs were associated also with the method we were testing? So here, a lower value means that the alignment works. What's the contrary of better? Worst. So a positive diagonal matrix works 0.35. A positive group matrix works better. Randomizing per row or fully doesn't make a good job, while the real matrix, the one calculated as I described you a few slides ago, was very efficient and strongly helped in the alignment of the RNA secondary structure. So in summary, the bare encoding allows us to compare structure with low complexity, and also thanks to this definition, the construction, the building of these matrix, substitution matrix for secondary structure element, in the comparison, we can also include evolutionary knowledge. So we have built these tools which are available as servers. For the global comparison, there is the bigger tool. And one can use this tool if wants to establish the association between full sequence and the servers allow the comparison of one RNA molecules versus many, or many molecules versus many, or one RNA versus precompiled data sets. They have to be precompiled because we have to take the time to fold each one of the molecule. So there are these 3' UTR and the long non-coding RNA data set of Homo sapiens and Mus musculus. And it is the output. One can have these pairs of RNAs with the evaluation of how good the alignment is, and each one of these pairs can be opened, and you see the alignment of the two molecules and their secondary structure and how they can resemble one the other. About local comparison, we developed this other method, I will tell you more later, which is called BEAM. The scope of BEAM is, as I was saying before, the analysis of a high number of different molecules. But when I say high number, I mean tens of thousands of molecules that have to be folded and analyzed in search for common motifs, which are present in at least a certain number of them. So with this method, one can analyze the RNAs, I will show you then later, RNAs identified in CLIP experiments. One can analyze homofunctional RNAs, for instance, RNA editing examples, to see if there are common 2D motifs which can help identifying what happens in these molecules. Potentially co-regulated 5' and 3' UTR, for instance, to identify sites of interaction with proteins acting on them. Beam use a scoring function. I won't go into this detail, but this I just wanted to show that there is an objective function that describes an alignment between different windows located in the different molecules, and this scoring function is optimized using simulated annealing. So this is the form of the Beam algorithm. It takes in input a number of RNA sequences, as I was saying, up to 100,000, and secondary structures. And it starts randomly selecting windows here and there in part of this secondary structure and sequences, and each alignment is evaluated and then sent to a perturbation machinery, which adds windows, remove windows, shift windows, resize windows, and realign. And it looks for a convergence of a score after many runs, and when it identifies a motifs, it applies a mask on the identified motifs, and it goes on looking for new motifs to be identified. So the output of this method is composed of sequence motifs and of secondary structure motifs. Now, there is a big problem working with this data, and the statistics is not easy to handle. We have scores which are calculated by beams, and that depend on how much the alignments identified are nice and conserved, but how much really they are statistically significant. This is another point. So we didn't know how to act, and this was the final decision, which is not perfect. But if you have other suggestions, we would be happy to think together. So we took all our FAM, this is this famous database of RNA molecules, and divided this big database in bins according to the length of the different molecules and the structure of the different molecules. So the bins were defined so that each bin could contain approximately 2,000 different molecules. And when we had the data about one RNA molecule, then the background that was used was its bin in our FAM, and we calculated the zeta score, and that's it. So in Beam, we can work to identify sequence and structure motifs, and we can calculate also the concurrency of the different sequence and structure motifs and the distance on the molecule that we analyze with Beam. Because if something is more conserved, if distances are somehow conserved, this can be an interesting hint highlighting the importance of what has been found. Now, apart from the calculation of the statistical significance, which is an issue, we have other issue. Remember that what I said at the beginning, that the prediction of a secondary structure is far from being perfect. So how can we calculate the performance of Beam? Beam works with RNA secondary structures predicted with state-of-the-art methods. So, of course, its ability to identify RNA motifs depends at first upon the structure prediction accuracy. If the prediction is not good, then there is no way for Beam to identify a motif. And then we want to analyze high throughput experiment, as I was saying, clip experiments, where the experimentalists are flooded with thousands and thousands of molecules, and most of the data are just noise. These results, as is written here, are intrinsically noisy. So we want to evaluate, can Beam handle this problem? And how can we evaluate Beam ability to identify an RNA structure motif in a noisy data set? So two problems: structure prediction accuracy and noise. So we decided that a way to see if we were able to handle these problems was to work with artificial data sets. So we started from a true data set, which was composed of 10,000 unrelated RNAs, in which Beam had not been able to identify one single motif. And we started from these 10,000 molecules, and we decided to use a sequence, small sequence extracted from our FAM, that could be folded into a gold structure, and use it, this gold sequence and gold structure, to build known data sets. So these gold sequence and structure, gold sequence at the beginning, were injected into a certain number of molecules in this data set with different percentages. So in 10% of the data set, 20, 30% of the data set. And part of the data set was left without the injection of this gold sequence because it represented, we wanted it to represent the noise. Then we could go on with the folding of each one of these molecules. And of course, we had the different cases. In some cases, the gold sequence could fold into the gold known structure. In other cases, according to different accuracies which we know exist, it could not. So in this building these data sets, we could have everything under control because we knew what was the percent of sequences with the injection of the gold sequence, and we could calculate the accuracy of the folding, checking it after the folding. So we could ask this artificial data set how many of the sequences with the gold sequence have the correct gold structure. And Beam can identify motifs, and since we know everything of this data set, we know which ones and how many are the true positives, false positives, true negatives, false negatives. And out of this data, we can calculate the Matthews correlation coefficient and have a measure of how well Beam can handle these two problems, the noise and the prediction accuracy. This test, of course, has not been done with one gold structure or with one noise level. We used 100 different gold sequences injected and 10 different noise levels, 10, 20, up to 100. So each noise level dataset was created 100 times with random insertions. And this is the result. Now, first of all, let me tell you what is in the axis. So here on the x-axis, you can see the percent of the insert in how many molecules was present the gold sequence, 10, 20, 30, 40%. And here is the Matthews correlation coefficient. The higher, the better. Now, why we see four curves? Because we have divided the performance of the method according to the different accuracies of the prediction. In this way, separating the contribution of Beam as being able to identify motifs from the contribution of the problem of the prediction program. So in blue, you can see the performance for the lower bin of the prediction accuracy, the bottom 25%. Then from 25 to 50%, then up to the top mean is here in my. What is possible to see here is that when the noise is up to 80%, BEAM can identify a motif. And it's still, and it works. It identifies the good motifs, and it identifies it as soon as it is present in at least 20% of the data set. Now, about the other problem we were talking about, the one which has to do, which has to handle the problem that in a cell, not always the best structure is true, but also suboptimal structures can be used, can be the good ones. So this has to be implemented, and we are working on this problem for the BEAM 3.0 version. As soon as BEAM has one sequence, can use multiple prediction, not only the MFE, but also other suboptimal predictions. And in this way, increasing the probability of including the good prediction inside the story. And as you see here, we can have different RNA molecules, and in some cases, a motif can be represented into the minimal free energy structure. Oops, sorry. In some cases, the motif can be present in a suboptimal structure. So now the model considers only one possible suboptimal structure at a time for a given molecule, and optimizes the one that fits the motif model best. As I said, the motif can be at most on one of the many structures, of course, for each RNA. And now, the rest of my presentation is about applications. I would like to know if it would be worth making a pause, or if I can continue with the applications.
It is up to you. I don't know if there is some question maybe at this point.
Is it necessary? Because I have a general question, but I rather ask it at the end.
Okay. So go ahead.
Okay, go ahead. I'm not tired. If this was,
okay.
No, but I wonder if you are tired.
No.
Okay. This is my work, so I like it. But so applications. We have built all this nice castle of methods, of ideas, the alphabet, the substitution matrix, the different methods, but the final scope is having biologically interesting answers. So what were the ideas we had to exploit this method? First of all, of course, the first application possible has been to analyze clip-seq experiments in the different versions: par-clip, I-clip, E-clip, different cases. Then I will say a few words about what is a clip-seq experiment, just for the few in the audience that do not know. Then, through the analysis of several data of proteins interacting and binding with different RNA molecules, we identified RNA protein binding motifs in sequence and in secondary structure. And these motifs are being saved and stored into a database. And this database can be used then to predict the ability of a new RNA molecule to bind those proteins. Then what we did was to analyze known cases of interaction of microRNAs with their mRNA target, because as you will see, microRNAs interact with their target using just a short sequence, 6 to 7 residues long, and this doesn't seem much in a cell. Of course, there are cell compartments, but anyway, we decided to check to see if there were other signals that could help this specificity of interaction. I'll show you the results. And then also this last project is my favorite one, but it's still not finished. It's in progress, but I wanted to talk about it nevertheless. It's about the analysis of London coding gene structure. As I will show you better with the slides, it is known that eukaryotic genes can have evolved from smaller genes that could initially code for what now we call protein domains that could be rearranged in evolutionary time using what is called exon shuffling to build the nowadays eukaryotic gene, which is essentially and very often modular. And now with long coding RNA genes, this is true. Can we use this idea to be able to identify exons which are important functionally? I'll show you. So a few words about these CLIP experiments. What is a CLIP experiment? In their different versions and declinations. A CLIP experiment is done in this way. There is a protein, and on these proteins, very many different RNA molecules, a transcriptome is launched. And of course, some of the RNA bind to the protein, some not. Some are specifically bound, some not. As usually happen in experiments, you have true positives and false positives, but when you wash away the most of the unspecific RNA, usually it is used the UV cross-linking, and then the molecules are purified, digested, and transformed into DNA, reverse transcribed, transformed into DNA and identified. So out of an experiment of CLIP, you recover a very high number of initially RNA molecules, quite short, but most of them should be associated to the ability to bind that protein. Okay, so these experiments are available into a database or also another database. We took 100 of each CLIP, par CLIP experiments from the Dorina database and more than 200 eCLIP experiments from ENCODE. Putting all this data together, we had the data about 190 unique proteins and 70 protein domains. So for each experiment, we had from 1,000 to 20,000 short molecules of RNA listed as possible binding sites. Therefore, using bad tools utilities, we could enlarge these short molecules, 100 nucleotides at the 5', 100 nucleotides at the 3', to have folded molecules, because folding 20 nucleotides is considered not to be interesting. And of course, we know that also in this case, one can have better or worse predictions. So, as I said, we extended the binding site at both ends, and we filtered out the targets which were too long, because it is known that the prediction of too long molecules is not reliable, and decided to try to associate motifs to each one of these proteins and protein domains. I show you one case. This is the Slbp protein. The Slbp protein is involved in histone pre-mRNA processing, and it is already known in the literature to be able to bind a stem loop structure. So we tested the ability of Beam to recognize it, and it does. And Beam recognized this is the sequence motif, and this is the secondary structure motif. And about the coverage, the sequence motif was present in 66% of the molecules, while the structure motif was present in 59% of the molecule. And both motifs were present in 51% of the data set. This is the hypergeometric p-value, which is significant. You may dislike these figures because they are not that much, but please remember that first, these experiments are very noisy, so there is a background. Second, there is the problem of the prediction of the secondary structure, which we have to handle. So, of course, this number is expected somehow to be lower than this other number. And in general, both numbers are expected to be lower than 100. But anyway, this was the result with one single protein, and it reconstructed the solution which was already known in the literature. And this is the table reporting the known data. We selected the proteins here, which had already known sequence or structure motifs at the basis of the binding, and we were able to recognize those motifs. But also from these big data sets, we could have many more motifs in sequence and in structure that we could associate to the binding with many other proteins. So from this analysis, we could identify 2,500 sequence and 2,300 structure motifs associated with the binding of 186 unique proteins or 69 protein domains. So we could define this database, which is a database of RBP, ribosome binding protein binding motif. And inside this database, we have, for instance, this is a sequence motif associated to the protein FUS. We have the structure motifs associated to the protein Slbp. Other structure motifs, other sequence motifs, each one of them is associated to one protein. And so this is the server which is now submitted, the BRIO server. BRIO stands for Beam RNA Interaction Motif Search Tool. You can give a number, one RNA molecule or more, then the server folds these molecules and compares the molecules with this data set, with the sequence and structure motifs associated to the different proteins. So the motifs shared are identified, and the output is this molecule or these molecules could possibly bind to this protein or this other protein. And at the moment, a tool like this in the landscape of available tools for biologists working with RNAs is not present. So we hope. Okay, as I was saying before, we decided also to work with microRNAs. MicroRNAs are able to bind these few nucleotides, usually at the 3' end of a messenger RNA, and they bring the AGO protein in the RISC complex to interact with this mRNA. And at this point, the messenger RNA can have two different fates. One is the translation is stopped, waiting for the interaction to be released. And the other possible fate is the RNA is degraded and vanishes. We could analyze the results of a CLASH experiment. CLASH stands for cross-linking, ligation, and sequencing of hybrids. is a technique which is able to pair with a certain certainty one microRNA with its target and with its targets. We had 14,000 mRNAs in this experiment, and we decided to see if around in the mRNA we could be able to find other signals in sequence or in secondary structure which could explain this specificity of interaction, which seems really to be not too tightly bound to this relatively low number of residues, complementary residues, which don't need to be all complementary. So we analyzed the 380 microRNAs and all the targets from this published CLASH experiment. And of course, we had different results. So here, I like this graph. This is called the ball graph, but what does it mean? I try to explain. So it's relatively easy. On the x-axis, you have the significance of the structure motif. On the y-axis, you have the significance of the sequence motif. So since what we sign here is the minus log 10 p-value, then good things are here at the top right part of this graph. Then the different ball represents different microRNA. And the size of the ball is associated to the number of targets, and the color is associated to the coverage of the motif identified in the data set of the targets of a given microRNA. Okay, the balls which are rounded with red are the ones which we decided that were more interesting and decided to analyze. And this is the table of the results. So we could always identify a sequence motif, and in all cases, the sequence motif was the seed sequence. No other sequence motif was identified. And in many cases, we could identify a structure motif, which is not too dissimilar in all analyzed cases. There is a stem. So we can suggest that this stem can be involved in the interaction of the messenger RNA with the risk complex. And now the last part of the application, the one which excites me most, but that is still at the beginning, as you shall see. So we are searching for, as I was saying at the beginning, for functional domains in long non-coding RNA genes, reminding what is at the basis of the eukaryotic coding gene structure. As I was saying at the beginning, there are several genes in eukaryotes which are built using a combination of different domains. So here you have a gene composed of different domains, many of which are epidermal growth factors, EGF domains, fibronectin with multiple finger exons, plasminogen with the Kringle domain. And there are genes presenting one or more copies of the different domains. So as I was reminding you at the beginning, this could have happened with evolutionary time with the shuffling of the exons, which could help the evolution of the final eukaryotic gene. So exon shuffling in evolutionary time can have shaped the genes by spreading exons characterized by a stable structure and the function in different genes. Here you see about coding genes, the case of Sh3 domains. Sh3 domain is able to bind the phosphotyrosine. And in the human proteome, we have, I don't remember how many, but hundreds of Sh3 domains. All of them can bind a phosphotyrosine, but they are not all identical. They are different because each one of them can recognize the phosphatidylate in a different context. So this is clearly one of the games of Jacob and Monod of the evolution. The evolution can duplicate and then create variability, and the variability becomes useful. And in this case, so the same domain has been duplicated several times, spread in different genes, variated in several positions, so that these different domains can recognize different phosphotyrosines in different proteins. The same game is done with Sh3 domains. These are just two examples, but are very present in eukaryotic genes. So also Sh3 domains are present in hundreds of copies in human, but also in other genomes. And the common theme is that Sh3 domains can recognize polyproline 2 structures in proteins, which are these triangular peptides with different prolines. But also, like Sh2 domains, Sh3 domains are different, and the different residues which are present in the binding pocket allows the recognition of different proteins containing the polyproline 2 peptide. So you have similarity and variability. The similarity accounts for the stability of these proteins because these are stable and share the ability to recognize something important, a phosphotyrosine, a polyproline 2 peptide. And then the variability allows the proteins to recognize those targets in different cellular contexts. So the ability to fold into a stable structure is very important because if not, evolution should go and look for a stable structure for much longer times, and this is not the case. The same, we were wondering, could have happened in long non-coding RNA. So we decided to analyze the structure of long non-coding RNA genes, seeking for a modular organization of their exons. If we can find domains in exon sequence or structure present even with variation in different genes, then we may presume that they have a role and a function. And this is, we don't have yet the answer, but I show you the top of the iceberg of the first answers we already have. So we started from the human genome in Ensemble and started from a list of 35,000 exons of long non-coding RNA genes. Most of these exons contain repeats, very repetitive sequences, and this is very bad when one wants to identify similar sequences or secondary structures, because then you find an association, a strong association on repeats, and these are not interesting. So we decided to concentrate on the exons without repeats, which were less than half of them. Then some of the exons were overlapping and some not, and we decided to concentrate again to have a simpler data set on non-overlapping exons. So we ended up with only 20,100 non-overlapping, without repeats, long non-coding RNA exons. And what we want to find within these exons, we want to know if we can identify cases where, for instance, you have two genes with two exons which are similar, but they are switched. The one that was first in a gene is second in another gene. And this is one case which is interesting. Another case which is interesting, if we find domains, exons, which are present in two different genes and also are present in a third gene. So this is interesting. And this also interests us if we have the same exon present in more genes. this is an error in animation. Now this had to appear. We are interested in all these cases and also in combinations of those cases. So we took all these exons and started comparing them with each other. We started with sequence comparison, and this is what we found. Now you see different networks. The nodes represent long non-coding RNA genes, and if two genes are linked with an edge, this means that they have an exon which is very similar and is on both genes. Okay. Now I show you the results. Some results are too complicated to be viewed easily, but this is nice and not too complicated to see. So we call this network the star. And here is the situation of the different nodes involved in that network. The different boxes with different colors represent real exons. And the same color identify the same exon present in different genes. So this red exon is present in many of these genes. The blue exon is present in 2 of them, and the green exon is present in 3 of them. So this is interesting. Somehow could be one case of exon shuffling. Now, what else one can do? Try to understand what is the role of these exons, for instance. These are the links which have been found. Not all of the links have been identified. For instance, this is not present because they are not identical, of course. So the system is not complete. So we went to see if we could identify the similarity to coding exons, and the red exon has a similarity with one coding exon, the other one no. We can find matches with promoters, no in one case and yes in another, and more work still to do. The biggest work which is missing, of course, at the moment is the comparison done with secondary structure, but we have still to start it. So future work with this project is to include secondary structure as criterion in the search for similar exon, as I said. Check synthetic regions and orthologous genes to see if we can see that the situation is conserved also in other genomes. Repeat the analysis in other genomes to see what happens. And now conclusions. So I show you that we defined an alphabet for describing RNA secondary structure along with its sequence. I've shown you the alphabet and its strength. And one of its strengths is not only the ability to be fast in the comparison of secondary structure, but also in being able to support the building of a substitution matrix with the allowed variation in the secondary structure of homologous RNAs. I showed you how these resources could help us developing tools for the global alignment of RNA secondary structures, for the local alignment of RNA secondary structure, that is, the recognition of functional motifs, and for the identification of protein binding motifs. And then I showed you a few applications. Now the acknowledgements, this is the people that worked at this of the projects I described you. For BIM3, we have a collaboration with the University of Florence of experts in AI, because we are not very experts, but we are becoming more experts than before. But anyway, they are more professional. And this is a picture that we have taken because like everybody, I think in the audience, we are working at home and we have these meetings with Zoom, with Teams, with Skype. Okay, I think we can work, we do work. But of course, we are longing for meeting again together with a cup of coffee around the same table, a slice of cake, and laughing all together, really, because now when one of us laughs, then the other cannot be heard because the voices are alternate. Okay, I thank you for your attention, and I'm ready to accept questions. Thank you.
Thank you, Manuela. Thank you for the beautiful talk. Now the discussion is open. I invite everybody to ask questions, to raise the hand. There is a question from Emilio Capriotti. Emilio.
I will ask a question if don't have students. So
but the students will add up. After you. Okay.
I have actually different question, but I will start with one. So the work that you have done in the classification of the motif is somehow complementing or completing the work that somehow was started a long time ago, that about the SCORE database and then no longer maintained. So in this sense, so is there somehow a sort of description what could be the possible motif that can describe all the RNA structure that are already known, or still this has to be somehow completed? This would be at least the first question that I think I'm interested to, because I'm not following recently the literature of RNA, so I'm a little bit back on this.
No, as I was saying at a certain point during the talk, for instance, think about proteins. How many databases are present, interesting databases with the data about motifs? Many. But for RNA, this is an almost empty world. So we decided that we wanted to complement what was already present with as many new tools and data as possible. Trying to provide about RNA, I didn't talk about the Vienna package, which provides a lot of tools and of methods for the analysis of RNA sequence and structure, prediction of secondary structure. But we are not collaborating with them yet. We would like to. And maybe now through what Rita mentioned at the beginning, this 3D bioinfo community, it is possible that we start talking. But of course, I think that, as I said, for RNA, there's really not enough to answer to all the many different questions which can be done. Mm-hmm. I hope I answered your questions.
Yeah, no, you answered. So just if there is another, maybe because I was more involved on the part about 3D structure of RNA, and yes, there is some overlap. So in the past, there was this RNA puzzle that was similar to CASP. Is this still active?
Yes,
or,
yes, it is still active.
Okay.
It is regularly done.
Okay, but they are also doing different challenges, right? Secondary structure, tertiary structure. There are many challenges there, right?
Yes.
Okay. So I can stop here if there are other questions.
Okay, more questions? Students? PhD students? In the meantime, Professor Calladio.
No, but I don't see the hand. The hand bottom.
In a reaction, but okay.
Okay, fantastic.
For everybody in the bar.
If there are students, I will shut up and wait.
There are no students by now, but you can go ahead.
No, I do have a very general questions, and perhaps forgive me for this question, but I'm not very much, I would say, in the field of RNA molecules. So I just follow the very principal foundations and whatsoever is in literature. My question is relative to the in vivo situation, in which you have these flexible molecules called RNA of different types, we know, and specifically also long coding, long non-coding RNAs. And then there is the problem of their interaction with proteins. So all these motifs of secondary structure and perhaps this organization that we know because we are studying RNA organization mainly in the solvent. So whatever we know because we ended up with even structures derived from, I would say, in vitro situations. Do you think that they are really, to be seen as a property of the RNA, or possibly this is due to the interaction with proteins? So when you have something flexible and you bump into something that is more stable, basically the electronic affinity is what dictates somehow the interaction. So I was wondering, since you identified, if I took the note correctly, some 69 protein domains that can bind RNA, then there is a specificity towards the RNA secondary structure that is, so to say, that each one of these 69 protein domains has its own preferred organization. And then if you substitute the sequence, perhaps, of the RNA, perhaps you will find the same motif, or there is something that is more general. So is the protein that induces the putative secondary structure that eventually is, is visible and you can find it because somehow it has been, how would you say, it has been recognized, it has been co-crystallized or whatsoever. I don't know if my question is...
No. Your question is terrible, but it's true. No, of course, because it's a big problem. Okay,
I would like to have your opinion.
Yes.
I want to have an answer.
I tell you, of course. These data, as you said, are done in vitro. These clip experiments cannot be done in vivo, I think. And so we have a partial view of what happens, but okay, partial, but in vitro those RNA molecules bind that protein. One thing that can be done and is done in vivo is the probing of the different molecules and the analysis of the structure at the single nucleotide level. And probably in vivo, the RNA, while is being modified by the method that is used, is interacting with the proteins. So those data tells us something about the structure of the RNA in vivo. And we can use those data to model the secondary or the tertiary structure for the ones who can do it of that RNA. So it is a difficult story. First, the problem of the prediction of the secondary structure, then the problem of interaction. Of course, an RNA molecule by itself will fold probably in a way, binding to a protein will fold in another way. So we have problems.
And the other story that I didn't follow at all is, at which stage are we when we think of messenger RNA? Is messenger RNA carried along because it is, it is bound to proteins, or it is somehow related to proteins that can help the messenger RNA to reach the ribosomes, or not? What is known in the field at the moment, since you are following it?
No,
I take advantage of your...
But I'm afraid I didn't understand your question. Messenger RNA, what people think is that messenger RNA is not structured. I don't believe it.
Yeah,
because
me too.
why? I mean, the messenger RNA, oh, I'm a messenger, so I stay linear. No, of course, it's crazy. And of course, anyway, in the nucleus, the messenger RNA is covered with the proteins which help
in the
the splicing. Then they bring, there are proteins bringing it to the cytoplasm, and then it is covered with ribosomes.
Okay, so we don't know too much about the interaction of messenger RNA with proteins in the cytoplasm.
What we think is that the secondary structure of a messenger RNA cannot be too strong, because if there were strong structures, and sometimes there are structures in messenger RNA, this means that, for instance, translation is blocked, or rallentata. How do we say rallentata?
Yeah, slow down
or slow down. Yes. So structures can be functional in the sense that a structure in a position of a message can mean that the translational machinery has to slow down at that point for some reason.
Okay, good. So hydrogen bonds, we know, can be, there are several other instances that can interfere with the stability of hydrogen bonding. So we don't know. And apparently the situation is still, in spite of Google, the situation is still unclear. And so we will have work to do for the next 50 years, perhaps. Okay. So there is a question. Did you notice it, Gigi?
Yes,
from
there is
Aldo
a
Vito, whom I don't know, and I thank him for the question in any case. So you are handling the discussion,
yes.
Gigi. Please go ahead.
So Aldo Di Vito, that is a PhD student, asked two questions. What about the protein affinity of the identified RNA motifs? What are the expected KDs? This is the first question. And the second, how do you manage non-canonical nucleotides and non-canonical base pairing, Uxtane, for instance? And it thanks you for the interesting talk.
Hey,
I don't know what is Uxtane.
Augustine, yes. The bind. No. It's a. So I was talking about in stems you can have Watson and Crick complementary pairs. Augustine bonds are the ones which are left free, the side of the bases which are left free from the Watson and Crick pairs, and can be used anyway to recognize nucleotides. And for instance, G quartets are formed with Augustine bonds and other structures in the cell.
Thank you, Manuela.
No, sorry.
Come and learn from you. Okay.
I'm in love with Augustine. I have a personal love for that bonds. But anyway, about the KD, this is a terrible question. I don't know. I'm only analyzing those data, trying to extract information from the experimentally bound sequences. So in that experiment, the one analyzed and present in Dorina or in ENCODE, somebody used a bait protein to try to bind as many molecules as possible, and some of those molecules were identified. Then your question is clearly interesting. If possible, I would like to have the answer, but I don't have it. And I don't think that also nor experimentalists that did the experiment have news about the KD. They only know that in that conditions that those RNAs were bound, but the intensity of the binding, its stability, I don't think. The other question? No, I remember the modified nucleotides.
Yeah, the modified nucleotides and the non-canonical base pairing.
The nucleotides that are modified are out of our control. We don't know. There is at the moment no method, to the best of my knowledge, there is no method able to fold an RNA molecule with the modified nucleotides, taking into account the modification of the nucleotide. So it's out of our possibilities at the moment. Like pseudonodes. Again, there are a lot of we did a work for general purposes. Then, as is easy to say, the devil is in details, and details are the modified nucleotides, the strange secondary structure which are not somehow easily recognized from predictors and so on. These are clearly holes in the theoretical construction that we would like to be perfect, but it's not.
Thank you. We have a question from Professor Roberto Tuberosa. Excellent seminar. Thanks, Manuel, and congratulations. Not sure to what extent you and your colleagues are attempting to link this fascinating variability to phenotypes, mainly QTL in living organisms. What evidence, if any, is there that the dynamic expression of QTLs during the ontogeny of an organism is due to the changing structure of RNAs that you have described as a function of the internal and external cues?
This is a beautiful question. I would very strongly like to be able, no, of course, this is the final aim, to be able to link these different molecules to a phenotype. In certain cases, there are data, but we tried to collaborate with people working with diseases, for instance, with muscle diseases, to try to see if different secondary structure could be linked to different phenotypes. But at the moment, we don't have good results yet. I'm sorry. But there is a sea, an ocean of long non-coding genes to analyze, and unfortunately, not enough data to be able to link this data with phenotypes. Not yet.
There is an addition to the question. Do you think this might be a component of the so-called missing heritability?
Yes. Yes, nice idea. Thanks. No, I did not think before, but yes, this can be a nice idea.
Thank you.
One of the
many.
yes, of the many possibilities. One more.
Following up to the long non-coding RNA analysis, when you are saying that you are mapping exons in the long non-coding RNA analysis, from where do you take the exons, and which is your criterion? Because, you know, exons sometimes can, you know, be different in different situations. And I mean, the border of an exon sometimes is a little bit floating.
We took the data from the ensemble database, okay, the last version of the genome, and they have a certain definition of exons, and now I don't remember the answer, but of course also for long non-coding RNA genes, figuraccia, also non-coding RNA genes are subject to alternative splicing, yes. And I don't know what we have done with alternative splicing. I should ask to the PhD student working on the project.
A very complicated scenario.
Yes.
And a very interesting lecture, and also a very brave person to enter such a complicated scenario.
A very crazy person.
Oh, congratulations. My congratulations. By the way, I forgot to say that you have been the strongest, I would say, driving force to organize bioinformatics in Italy. Because, you know, you started with the famous Pontignano schools, and you ended up being the president of the Bioinformatics Society in Italy. But you are a wonderful scientist in any case. Okay?
Nonostante questo.
Nonostante.
All right.
You're right. Thanks.
Okay.
I'm still honorary president of the society.
Oh, you are honorary. Well, this means that you are quite old.
Yes, I am, unfortunately. So I cannot take the vaccine.
No, me too.
There is a question from Tamara Pertegella.
Yeah, good morning. It's always related to the exon of long non-coding RNA, if I may.
Yes.
You said that the definition of exon has been taken from the ensemble database. Can we suppose that the exon are the one that superimpose to the genes from which long non-coding RNA are, I would say, transcribed? Because I know that the long non-coding RNA for the majority overlaps with the coding sequences. Are we just considering those, or are we considering also the ones that come from intergenic region?
Okay, we took only the exon which do not overlap, which are a fraction of all exons. I don't know if you, I'm still sharing the screen.
Yeah.
Oh, sorry.
Yeah.
No. Ecco. So we have discarded three. What do you see? The presenter's view or
the view for the presentation? So
okay.
Not your.
Okay. So ecco, we have discarded the overlapping exons. Just to have a clearer situation to analyze. In this way, of course, we are not taking into account the good information which could be here, but also these exons, which are very many and that we discarded. I think that we could go and look for interesting information. But at the moment, just to be fast, to try to see is there something or not, we decided to work with less exons and risk to lose information anyway, but not to have a complicated life from the overlapping of exons with other genes or with the repeats. I hope I answered your question.
Yeah, thank you. And I have another one that I missed the beginning. Yes. About the constraint that you give to the software in order to build the secondary structure. I understood that the final outcome is the minimal free energy structure and that you start from the give free energy that were present in the table you showed us. But then you show a picture that shows also the local nucleotide dynamics through space neighbors, for example. But how do we take those information if we can't start from evolutionary data?
I don't know if I understood your question. Okay, the data from those experiments are available as constraints that can be used in the folding on the single nucleotides. So for instance, if from those data, from chemical probing, you know that a certain nucleotides is exposed, you discard all predictions in which that nucleotide is paired, and so on with all the available constraints, which have been taken in different situations, in vivo with some of the methods, and in vitro with other methods. And of course, those data also, like all experimental data and like all, are not perfect. There are not black and white solutions only, but also shades of gray. But okay, those data can anyway be used to arrive at a better prediction of the structure. I think that the figure of the analysis of the prediction of the 5' of the 16S of the E. coli ribosome was clear enough. Did I answer your question or not?
Yeah, thank you.
Oh, thank you.
Thank you. Are there more questions? We have 5 minutes if you have any curiosity that Manuela can answer.
But anyway, if you look for my name in the web, then you can also write me an email with further questions that you may have, or proposals. If you have ideas about why didn't you try that, we do it together. Rita, this is valid also for you.
Okay. Okay, so thank you, Manuela, very much for this beautiful talk, for this beautiful lecture. I invite everybody to join us tomorrow for the lecture of Roderick Guigaud at the same time, so 11 in the morning till 13. Thank you, everybody. Ciao.
So a lot of thanks, Manuela, and all my warmest regards and a big kiss. Okay.
Okay.
Ciao.
Ciao.
Ciao. Thank you.
Thank you. Bye bye.
Ciao.
Thank you. Bye.
Bye.